Thank you for joining me today for Parker Biology. We're going to be discussing another important biological molecule. Today's main focus are the lipids. So lipids are another class of large biologically important molecules, but instead of being a true polymer like carbohydrates were, lipids are a diverse group of hydrophobic molecules. Remember, we said a polymer was made up of identical or similar subunits, and lipids don't get to be quite true polymers because their subunits are so diverse. Now, the one thing that all lipids do have in common is that they are hydrophobic or repelled by water. This is true because they're made mostly of hydrogens and carbons, or hydrocarbons, and these form nonpolar covalent bonds. The most important lipids that we'll look at today are fats, phospholipids, and steroids. So, what is a fat? Well, fats are constructed from two types of smaller molecules, glycerol and fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. Notice that even though lipids are not true polymers, they are still assembled using dehydration synthesis reactions. We can see the hydroxyl group and a hydrogen being removed to release a water molecule. In a fat, 3 fatty acids are joined to a glycerol by an ester linkage, creating triacylglycerol or a triglyceride. Just like the covalent bonds in carbohydrates had special names, glycosidic linkages, the covalent bonds holding lipids together also have special names. In this case, they're called ester linkages. Now, I can remember this because triglycerides form a giant E, and the name ester linkage, it starts with an E. It works. In looking at this triglyceride, can you tell how many water molecules had to be removed to form this molecule? That's right, 3 water molecules were removed in dehydration synthesis reactions. It's important to remember that the unifying feature of all lipids is that they are hydrophobic or repelled by water. And the reason that fats separate from water is because of the uneven distribution of charge in water. This results in hydrogen bonding, which excludes the nonpolar fats. While fatty acid chains are often between 16 and 18 carbons in length, they can vary in length, or the number of carbons, and fatty acids can vary in the number and locations of double bonds. Saturated fatty acids have the maximum number of hydrogen atoms, which means that they do not have double bonds. Unsaturated fatty acids, however, do have 1 or more double bonds. Fats made from saturated fatty acids are called saturated fats. Remember, we said saturated fatty acids lack double bonds. Because there are no double bonds, this molecule is saturated with as many hydrogens as possible. Now, most saturated fats do form solids at room temperature, and most animal fats are saturated. On the other hand, fats made from unsaturated fatty acids are called unsaturated fats, or oils. These are typically liquid at room temperature, and most plant fats and fish fats are unsaturated. When we talk about our diets and we worry about how much saturated fat we're eating, the reason that's a concern is that diets rich in saturated fats may contribute to cardiovascular disease through plaque deposits. What happens is because saturated fats form straight chains, these chains are very easy to stack together, and when they stack, they form plaque. And that's what's going to lead to clogging of the vessels. Now, along the way, somebody thought it would be a really good idea to turn unsaturated fats, for example, vegetable oils, into saturated fats, so we could have vegetable spreads like margarine. They did this by hydrogenation. They added hydrogens to bring about a more solid texture. Now the problem is, and we won't get too far in depth with the chemistry, but when we hydrogenate, we end up creating trans fats. The trans configuration, notice, allows for the molecule to form a straight chain. And again, the problem with that straight chain is it can pack closely together, and if it does stack on top of each other, it will lead to plaque formation and therefore cardiovascular disease. And that really explains why there's such a push to eliminate trans fats from our diets. Now, other fats that we might have heard about in the news are essential fatty acids. And if we say a nutrient is essential, it means that we cannot make it in our own bodies. We have to consume it in our diet. And one of the really hot topics right now for essential fatty acids are the omega-3 fatty acids found in some seafood. And this is required for normal growth and is thought to provide protection even against cardiovascular disease. What do fats actually do for us? Well, the major function of fats is energy storage. There's actually more potential energy in the hydrocarbon tails than in any other type of biomolecule. Additionally, we store our fat in what we call adipose cells. And adipose cells aren't just for energy storage, they also work to help cushion our vital organs and to insulate our bodies. In addition to fats, another important lipid is a phospholipid. In a phospholipid, two fatty acid tails are linked via a glycerol to a phosphate group. We can see several different models, all of them showing us these two fatty acid tails along with this phosphate and glycerol head. So within our typical phospholipid, we see a hydrophilic head region along with a hydrophobic tail region. Now, when we add phospholipids to water, they self-assemble, they put themselves together. And what we see are the hydrophobic tails hiding out in the middle together, whereas the hydrophilic heads are able to associate with water, either on the outside or the inside of this structure. Now, this bilayer of phospholipids becomes really important, and this is the characteristic arrangement of our cell membrane, the phospholipid bilayer. So we've looked at fats, and we've examined phospholipids, and the final main group of lipids are the steroids. Steroids are characterized by a carbon skeleton consisting of 4 fused rings. An example is shown here, cholesterol, and this is a really important steroid because it helps stabilize our cell membranes. Even though cholesterol is essential in animals for our cell membranes, high levels in the blood can contribute to cardiovascular disease. Now, the region up here can vary, and different steroids are created by adding different functional groups. Remember, structure determines function. So depending on what's attached to the rings, steroids can have very different functions. For example, both estrogen and testosterone are steroids, but their different functional groups make them very different hormones. That concludes our discussion of lipids. Thanks for joining me at Parker Biology.